Hello, can somebody please confirm you can hear me?
I can hear you.
Thank you. Let me get it up here. Boy, it doesn't look like I've got my highlighting on this one either. I don't know what happened to my highlighting. Let's see if I got it elsewhere. It doesn't have it either, so I'll have to play it by ear. Okay. All right, before we begin, are there any questions? No questions, so let's begin. Let me share my screen. And my hair looks something funny. Yeah, that's just the way Zoom is going, I think. Any questions before we begin? Sharing my screen now. You know, we're doing Lab 01 today. The ubiquity of microorganisms. Remember, you need to take your online plagiarism quiz by 11:59 p.m. this Saturday. You shouldn't wait till the end because if you have to take it over again, you may need a little bit of time to do that. You must achieve 100% on the plagiarism quiz, or you will get 0 on your infectious disease project. You should read about the infectious disease project in a file called Project Instructions in Campus on Canvas. Let me see if I can get that. I'm not in Canvas because I can't get to it. Zoom from there. Oh, good. The lecture has finished formatting, so my computer will work faster now. So if we go to our Canvas main web page and scroll down to, I think it's lab week 2. I got the infectious disease research project. Take a look at that. You should read it over before I begin talking about it. You will need to know how to do the infectious disease research project. And I will talk about that next week in the lab. This assignment is worth 100 points. It's due on August 19th. Any question about any of that? All right, if there's no questions, let's move on to Lab 01.
I know the infectious diseases says first come, first serve for your pathogen. When should we send you an email about that?
I'm not going to collect emails yet. So if you send it to me now, I'm going to just tell you, you're going to have to wait until the project starts. The project is going to start on Tuesday. So you can send me an email telling me what you would like to do your project on starting Tuesday. Okay, good question. But at the moment, I'm not going to start the project, so I'm not going to try and keep track of that. So don't send me the email until Tuesday, because the project hasn't started. All right. Somebody obviously looked at that. And Lab Module 1, Ubiquity of Microorganisms. There is some reading to do for this lab. So read about culture media, page 157 to 162, and obtaining pure cultures, just really a page 162 to 163. There's some supplemental materials in this lab. There's some video clips. The learning objectives for this lab: 1, know that microorganisms are almost everywhere. 2, describe the purpose of culture media. 3, describe what are colony abundance and colony morphology. And I wonder if this is the one where we talk about colony diversity. I think this is the lab we'd also talk about colony diversity, and I should have that up there as one of the objectives. Anyways, define the following terms. What's inoculation? Ubiquity of microorganisms. What's something that's sterile? What is isolation? What is incubation? What is in vitro, like in vitro growth? What is a culture media? What is a broth? What is an auger? What is a slant? What is a deep? What is a fastidious organism? What is a non-fastidious organism? That's not up here, but you should know that. And then what is non-selective media? And what is ubiquitous? And what is a colony? Any questions about any of that? All right. Microorganisms are organisms that are too small to be seen with the naked eye. This does include viruses and prions, which are put in this group, even though they're not really an organism. They do not have cells. You could even argue, are they alive or not? Or at least viruses. We're also going to include helminths in this group because microbiologists have studied helminths. And the adults of the helminths are usually not microscopic. However, the infective stage for when people become infected usually is microscopic. And because microbiologists have studied organisms that cause disease, and the helminths are definitely an organism which causes disease. You should know the phrase ubiquity of microorganism refers to the concept that microorganisms are essentially everywhere. In our everyday experience, microbes are virtually ubiquitous. They're in the air we breathe, the foods we eat, the water we drink. They're on the table that we're going to be pretending to spread our microorganisms that we're growing. And this is a problem for microbiologists because when we're working with one species, we usually do not want it to get contaminated by microorganisms in the air or on the table around us. And the reason for that is it's much more difficult understanding the results of a test if there's more than one organism present. For example, if the test is positive, is that because of the organism that we're trying to study, or is the test positive from some other organism which is contaminating our sample? Humans only have microorganisms in a few places. We do not normally have microorganisms in our blood, in our cerebrospinal fluid, in our muscles, bone, and nervous tissue. We actually spend a lot of energy with our immune defenses to keep the body, internal body, clean of microorganisms. We call something sterile if it is free of living cells. In the lab, if you were to come into a real microbiology lab, we would give you sterile media. And how we make that media sterile is we put it in an autoclave. And then it's cooled, and then it's given to you. Obviously we'll be doing everything online, so we'll just pretend that I'm going to give you media and that you're going to grow organisms, and it's just pretend. But that's what sterile means. You're also going to learn some basic lab protocols, such as how to use solid media, how to label cultures grown in a microbiology lab, and procedures for disinfecting lab benches and for washing your hands. All right, culture media is something that we grow microorganisms in. A culture media, also called a growth media, will be given to students if we were going into a real microbiology lab, which would be sterile. And the medium will have all the nutrients that are required for growing the microorganisms of interest in the lab. So most culture media would be able to grow any non-fastidious microbe. Non-fastidious microbe means that it does not need special additives to grow. Something like E. coli would grow on essentially any culture media that we would give you in the lab. There are different types of culture media. If it's a liquid culture media, this is called a broth. And it's commonly put in a test tube because if you had it in a plate, the liquid would be too easy to spill out, and then you get contamination and microbes growing in your culture. We also have solid media, and it's usually solidified with agar. Agar is a substance extracted from seaweed, and it's used to thicken the broth and then solidify it. Agar, if you're not familiar with baking or cooking, is a substance that's sometimes used to thicken whatever people are baking or cooking. We'll talk a little bit about that. You can thicken the broth and make it a solid without using agar. Usually agar is used, but you could add starch or you could add gelatin. The problem with gelatin and starch is that some microorganisms can digest those ingredients. And if you start digesting the gelatin, instead of having a solid media, you will start to get a liquid media. And the liquid will usually be on top of the solid agar, or solid media, because it's media, sorry. It's not agar because we have Jell-O in it to make it solid. All right, so when you're doing this lab, you should watch this little video on preparing pour plates. I'm not going to go over it now, but you guys should watch it when you work on the lab. Here we see a tube containing liquid broth, which is a liquid media. Here we have two solid media. This is called the deep, where the agar is just put across flatwise. And this is called the slant, where the agar is put at a slant. And that is usually a agar plate, so a media on a plate. And plates are almost always solid media. As I stated, if you had a liquid in there, it'd just be too easy to spill it. We have different types of media. Today, we're only going to talk about one type of media, and that is a general purpose media. A general purpose media is non-selective. It usually has complex composition, and it supports the growth of most non-fastidious microbes. They will use the peptone or the yeast or the beef extracts to grow. There are 3 commonly used general purpose media in microbiology. And if you were to actually come into a microbiology lab, you would probably use all 3 of these. The first is nutrient agar with yeast extract, abbreviated NA, and it has nutrients from beef and yeast extracts, as well as peptone. Tryptic soy agar has peptone from soybeans added to it. And does it have any other additives? I don't remember if that has any other additives that they would, microbes would eat. Nutrient agar is just peptone extracts and beef extract. So you get the cow and grind it up, and then you add the water, and that's all it is. So the NA is a little richer than the nutrient agar because it has added yeast extracts. So these provide nutrients for most microorganisms like E. coli. Any organism that's non-fastidious generally will grow on any general purpose media. Another reason for using general purpose media is they are cheap. And so that's why we use general purpose media. There are other categories of media, and we will discuss them in a later lab. Whenever you're working in the microbiology lab, you should appropriately label your plates and your tubes. How do you appropriately label them? You put 4 or 5 items on the plate or tube. You put your name, and that could be your initials. You put the date. You put the type of medium it is. Like if you're growing your culture on NA, you write NA for the type of media. The incubation temperature that you grow it at is optional. And then you put your inoculum, and that could be what you're growing, or it could be what you expose the plate to. You just say what you're growing or what you've inoculated. On a test tube, you normally write that all in a piece of tape and then attach it to the test tube. On an agar plate, you write it on the bottom of the plate and you write it around the edge of the plate. So Travis is the name of this individual. E. coli is what they're growing. This is the date. I can't read that, but it's probably 2009 or something like that. And then the media is TSA. And then the temperature is optional. And this person has chosen to put that on there. And it was grown at 37 degrees Celsius. Come on. So when we're using cultures, when we first put microbes in the culture, that is called an inoculation, the placement or transfer of microbes onto a sterile culture media. You can do this with a variety of tools, and we'll talk about some of those tools today. Incubation is then placement of the culture media, the container that you're growing the microbes, in an appropriate environment to grow the microbes. Usually we incubate our samples in an incubator, and we have simple incubators that all they do is increase the temperature that the microbe grows at and add humidity to the air so that the plate or culture will last longer and not dry out quickly. There are more complicated incubators, and a more complicated incubator can control how much oxygen is present in the incubator, how much carbon dioxide is present, as well as the temperature and the humidity. And I forgot to mention that we also can control the time, and that is we usually grow cultures overnight or in a real microbiology lab. We frequently start them on Tuesday, and then we'll look at them on our next lab on Thursday. So let's look at an incubator down here. So this is a complicated incubator. This dish has water in it to provide humidity, and it's heated either 37 or 35 degrees. So it does warm the temperature, which most bacteria grow best around 37 degrees. And this one can control the oxygen and the CO2, which actually in our microbiology lab, we don't have any fancy incubators like that. Come on. I'm having trouble with my mouse. The incubation time will vary on what you're growing. We usually don't grow Mycobacterium, but if you were to grow Mycobacterium, it tends to grow slower than most other families of organisms, and you may need to grow it for more than 2 days, for example. All right, when we incubate agar plates in the incubator does warm the plate. And since the agar plate has agar in it, there's moisture in the agar. The plate with the agar, I don't know if you can see this, hopefully you can, but moisture will evaporate from the agar, and where it will go is on the underside of the lid. And so droplets of water will start condensing on the lid. And with time, the droplet will get big enough that the droplet of water will actually drop from the lid onto the top of the agar surface. And that will not necessarily contaminate the plate, although it does increase the risk of contaminating the plate a little bit greater. But if it does drop into the plate is sterile. The lid should be sterile. The water should be sterile. The only thing that's not sterile would be the edge of the lid. So if the water were to drop down on the edge, it's actually going to drop outside of the plate. So that shouldn't be a problem. But if you take off the lid and then move it like that, the water could go down the edge and then drop into the plate. And so it does increase the risk of contamination, but it's not contamination we're worried about. When you have water droplets dropping from the lid onto your agar surface, that'll disperse the colonies you have and making a mess of the colonies you're growing. And so to solve this problem, whenever we have an agar plate, we stick it in the incubator upside down. So now the agar is on the top. And if moisture were to come out of the agar, it will condense on the agar, and the agar would disperse that droplet, so there'd be no drops found. If for some reason the droplet were to go on the lid, who cares, because there's no bacteria on it. So whenever you're growing a Petri dish, it should be grown in the incubator upside down. Any question about any of that? All right. When we're looking at microbes after they've grown in the incubator, you'll notice that there are some differences between the colonies. Like that one, come on, is a different color from all the others. And it also looks different. It has an orange or yellowish ring around the main part of the colony. So this one would be, what would you call that? Multi-layered. These colonies could be the same, although they're different sizes, but that one looks a little different. The color looks a little different, and it's smaller than even these smallest colonies here. And that white colony is clearly different. These two colonies are different. That colony is clearly different. So we have different colony characteristics and different colony colors and shapes, and that can give us some information about our growth that we're growing. I don't know how else to word it. It tells us some differences, like on here, we have, what would we say? 1, 2, 3, 4, 5, 6, 7, at least 7 different types. Actually, I think that one, well, that one could be the same as that one. At least 7 different types of colonies growing on this plate. So there's at least 7 different species growing on this plate. Any question about any of that? There are some differences between bacterial colonies and fungal colonies. First, fungal colonies tend to be bigger than bacterial colonies. And let me blow that up a little bit. That one's obvious, but fungal colonies are not clearly distinct on where the boundary is. It's kind of fuzzy. Even this one's kind of fuzzy. It's fuzzier right there. And then when we look at a bacterial colony, their boundary is very distinct. It's very clear that there's growth and right there is not growth. Okay. Whereas right here, we got a little bit of growth and it's hairy or fuzzy. So the fungi grow hairy or fuzzy, and there tend to be bigger colonies than the bacterial colonies. We can make other characteristics on the colonies. Let's go to the table of that. Colony characteristics. They have a different shape. There are circular colonies, rhizoid colonies, irregular colonies, filamentous colonies, and spindle-shaped colonies. The margin is the edge of the colony. We have an entire margin, which would be something like in a circular colony. We have the edge, which is undulate or lobate or curled. And I'll never ask you, is this colony margin undulate, lobate, or curled? I'll also never ask you, is this colony margin rhizoid or filamentous? However, you should be able to distinguish between a rhizoid or filamentous and an undulate or lobate or curled margin and an entire margin. To see the elevation, you normally have to take the lid off and then look at the colony from the side. Is the colony flat? Is it raised like in a mesa? Is it convex? Is it pulvinate, and that's super convex? Or is it umbunate, mountain shaped? Right here. And actually, I think I see an umbunate right here. I think this one's an umbunate. That's why it's got 2 appearances. Right there would be the center of the umbunate, and there would be the edge. But I'd never ask you, is the elevation of the colony convex or pulvinate? But you should be able to tell, if you can see the colony, if the colony is flat, raised, or one of these 2, or umbunate. The size of the colony is something like the size of a pencil, and that is, a colony is punctiform if the size of the colony is smaller than the tip of the pencil that you write with. It's small, the colony is small if it's between the size of the tip of the pencil and the size of the Let me think about that. The size of the eraser. We used to have this. I'm gonna have to think about that. If it's large, it's larger than the size of the eraser. So if it's small, it's around the tip of the pencil. And then moderate, it's someplace between the eraser and the tip of the pencil. And punctiform, it's just a pinprick, smaller than the tip of the pencil. Any question about any of that? The texture is a problem that students have, and that is when they look at the colony, they can't tell if the colony is smooth or rough, but if you can tell, then it is smooth or rough. And let me just show you. Come on. So this colony has a smooth surface. This colony has a rough surface. And it's kind of hard to view that here in this photograph, but this one's clearly rough and that one's clearly smooth. The same thing with the glistening colony. This one is clearly glistening. This one is not. That one is not glistening either. That one is probably not glistening, but I'd like to actually look at it. That one probably isn't glistening either. I'd actually like to see it in real life to be certain of that, but so the colony appearance can be glistening, that means shiny, or they can be dull. The colonies can be pigmented or non-pigmented. If you can see a color in the colony, it is a pigmented colony. Come on. So all of these, which are yellow, are pigmented, including this one. This colony and this colony and that colony and this colony is not pigmented. Black is not a color, it's the absence of color or the absence of light. And so that is not a color. And then anything that's off-white, white, or tan is non-pigmented. And then you can classify the colonies on their optical colony. Are the colonies opaque, translucent, or transparent? Transparent colonies are very rare. They have the appearance like a drop of water. You can see right through the colony. Opaque colonies are the most common. And actually everyone I'm seeing here is probably opaque. Let me see if I can find a translucent colony. Oh, here. This colony right here on this part, that might be two colonies, one on top of this colony. It's hard to tell, but that could be a different colony from this one. This colony right here is translucent. You can see through at least part of the colony. So that would be a translucent colony, and that would be a opaque colony. Any question about any of that? All right, this on the edge looks like it's a translucent colony, but inside the colony, it's not translucent. All right, those are the different colony characteristics. You will be pleased to know that I'll provide you with this table if I'm asking you characteristics about the colonies. The only thing I'd expect you to know is something like circular and irregular. I guess I would expect you to know flat, raised, and umbunate. Smooth or rough, shiny or dull. And I do realize that students often get confused on what colony is smooth and what colony is rough. All right, so this is telling you some characteristics. Colony A, if I can get to it, is a very large colony, large, pale yellow. I actually don't see the yellow in that one. Circular shape, I would call it a regular shape because that's definitely not a circle there and there, although the rest of it right here is circular. It's shiny. That's hard to tell in this photograph. Pulvinate, that's impossible to tell in this photograph. And the edge is entire. That I would agree with. Colony B, this one will be easier. It's a medium-sized colony. Cream in appearance, meaning it's not pigmented. Irregular in shape, I agree with that. It's a rough colony. That one is hard to tell from this photograph. And flat, it's impossible to tell from this photograph. All right, when we're talking about colonies, it's important to understand two terms, and that is abundance and diversity. The abundance is simply the total number of colonies on the plate. So if two colonies look different from one another, they're probably different species, although you can't be entirely sure without doing further testing. But it doesn't matter because different species still give rise to the colony abundance. So here on this plate, like I said, we had like 5 or 6 different species for sure. And I don't know how many colonies this would be. What I'm going to do is say, this region right here, the colony abundance is 3, okay? I don't know how many that would be right there. If I go there above 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. And I'm not sure if that's a colony there. 13 or 14 would be the abundance for this region of the plate. Obviously, when you give abundance, you're supposed to count the entire plate, but that's a few too many for me to count. So I would say something like around 50 is the abundance on this plate. The diversity is the different types of colonies on the plate. And you have to look at the colony characteristics, the color, the shape, and the texture to identify the different colony types. For example, this right here is clearly different than that one right there, and then that pink one there. And it's clearly different than that one there just by the shape of the colony, as well as by color. Okay, there's too many colonies here for me to guess the diversity here, but this colony is very small, much smaller than those, so I'm going to guess these are different. Even though they have a similar appearance and similar color, they're probably different because of the size of them. So you have to count the total number of colonies to get the abundance, and then the diversity is the different types of colonies on the plate. In which case, for diversity, these two are clearly the same species, so you'd only count each of those as one. And then these 3, this one here, that one there, and that one there, are probably the same. And so you count them as a second diversity. Any question about abundance and diversity? Here's a real world practice. What is the abundance of cats in this picture? Come on, I'm not going to give you the answer. You got to supply it. What's the abundance of cats? I didn't hear you. Can you speak up?
Yeah, 7.
7. Yes, 5 cheetahs and 2 lions. And what's the diversity of cat species in this photograph?
2.
2, the cheetahs and the lions. All right, good job. Thank you for volunteering. You should know the terms of this lesson. And then we're going to work on the lab. Let me go ahead and talk about the lab. You don't need to go through the references. Let's go to the worksheet. Remember, when you're turning in the lab, only turn in the worksheet. It's much smaller than the lab module. And if everyone were to turn in the lab module, my Canvas website would eventually become full. And if it ever does become full, you students will not be able to submit an assignment. And I don't want that to happen. So only turn in the worksheet for each lab. If you turn in the lab module, the very first time I will give you a warning, but on the second time, I will apply a penalty. Any question about any of that? So please type your answers directly into this Word file, then save it and submit it to Canvas by 11:59 p.m. Week 1 Saturday. Remember, lab 00 must be turned in by 8 a.m. Friday morning, because I'm going to take attendance from who turns in lab 00. All right. So in lab module 1, we're exposing a plate to different environments, to the air, the lab bench, your hands, your lips, and the soil. In procedure 1, we're uncovering the plate and exposing it to the air for a given period of time. I think it's exposed to the air for 15 to 20 minutes, and then you incubate it in the incubator for 24 to 48 hours. And we're going to give you the picture because you can't obviously do this. Take an agar plate, expose it to the air, put it in an incubator, and then take a look at it 24 to 48 hours. So we've done this for you and supplied the picture. And you need to tell me, what is the colony abundance on this plate? What is the colony diversity on this plate? Now, your numbers do not need to be exactly the same as mine. As long as you're close, I will accept your answer. However, if you are far enough away, meaning you're far away, I will not accept your answer, and you will get the question wrong. Any question about any of that? So procedure 1A, what's the colony abundance? Procedure B, what's the colony diversity? And procedure 1C, when you are incubating a plate, when you're inoculating it, when you're inoculating it, what step would you take to reduce airborne contamination of the sterile agar on the plate? So we're going to pretend this is the plate, and you've taken off the lid, and then you start streaking on the plate. What steps can you take to reduce airborne contamination of the plate? Write your answer there. There's generally one answer, but there are a couple of acceptable answers. Okay. And it has to be something you're doing while you're inoculating the plate. So don't tell me about put the plate in the incubator upside down, because that is after you've inoculated the plate. Any question about that? All right, procedure 2 is, by the way, you do not have to answer the question on procedure A, what's in the air you breathe. That's just what procedure 1 is about. but you do need to answer these questions. Okay. Procedure 2, what gets on your arms when you put your arms on the bench? You don't need to answer that either. In procedure 2, what we're going to do is take a sample of the lab bench before it has been disinfected, and then take a sample of the lab bench after it has been disinfected. You just swab the top of the bench with a sterile cotton swab, and then put the cotton swab on the agar plate and streak it on the agar plate to see if you get growth. And here is the streaking of the lab bench, which would be the table you'd work at in the lab, showing growth on the plate. So these white specks are colonies on the plate. It's not very clear because I think this plate is upside down. There's agar between the colonies and our eyes. But anyways, that's the streaking of the table before you disinfect. And this is the streaking of the table after you disinfect. Oops, wrong one. The first question is procedure 2A, did table disinfection work? And you should explain yes or no. And then procedure 2B, answer the question, what is the purpose of allowing the table disinfectant to air dry on the table? So when we apply the disinfectant, we wipe it around and then get the worst of it off with paper towels, but we do leave a little bit on so that the disinfectant can air dry. So answer that question. Procedure 3, we're going to sample your hands. You do not need to answer what's on your hands. What you do is you sample your hands before you wash them and after you wash them. You're washing your hands appropriately with soap and water. And you do need to dry your hands because the water on your hands will wick the microbes on your hand to the agar of the plate. And the very first time I did this, the students were getting more colonies from their washed hands than they were getting from their dry hands. And like I said, the reason was simply because they didn't dry their hands, and the moisture from their hand was wicking the bacteria from their hands onto the agar. So you do need to dry your hands after you wash them. And the paper towels are generally good. Most paper towels do have some contamination, but it's very low. Okay. And so compare dry hands before you wash them, then go wash your hands, dry them, and then touch the plate. Here is the growth coming from 3 fingers, and this one it looks like 4 fingers. Before you wash your hands, and then the growth on your hands after you wash your hands. And yes, you dried them. The first question is, does hand washing work? That's simply a yes or no. And then 2, suggest one reason why hand washing did not result in the complete removal of microbes. Here's a hint. It may have to do with the normal microbiota on your skin, as well as what you washed your hands with. If you do research to get your answer, please provide a citation for where you got your information from. Students frequently get number 2 wrong, and that's because this is a thinking question. It isn't necessarily easy. You have to think to come up with the answer. If you get it wrong, I'll just tell you you got it wrong, and we'll point you in the right direction. And hopefully the second time you'll get it right. Any question about that? Procedure 4, you don't have to answer the question, what's on your lips or your tongue when you kiss your significant other. What we did with procedure 4 is we expose somebody's lips to a petri dish, and we expose somebody's tongue to a petri dish. And if you want to know who this is, it's probably Roberto Anatori, a microbiologist who's the lead instructor of microbiology at Clark. And I'm saying it now because he's not on campus. He's actually on sabbatical in Australia. And anyways, he probably did this. Please note the note above the tongue picture before you answer the question. And then, what is this, procedure 4? Procedure 4A, answer the question, what plate has higher colony abundance, the lip plate or the tongue plate? And B, why do you think this is the case, even though you might expect the abundance of both the lips and the tongue to be the same, because they're, quote, very close to each other, and you probably will occasionally lick your lips. Why might we see a difference between the lip plate and the tongue plate? There are 3 answers that I'm looking for, but actually students occasionally come up with a very good 4th or 5th answer, but there are 3 that I'm looking for. And to answer this question correctly, you must provide at least 2 of those 3 correct answers. And like I said, I do realize there's more than 3. There's at least 4. That could explain the difference. But you must come up with at least 2 reasons for why one plate has more than the other. And then C, most people get this one correct. Why was the tongue plate incubated at a higher temperature than the lip plate? I didn't talk about that, but the lip plate is, I think, grown at 37 degrees. The tongue plate is grown at 37 degrees, and the lip plate is grown at 30 degrees. Why did we grow them at different temperatures? Procedure 5, you don't need to answer this question. We're going to answer it with this procedure. And are there microorganisms in the soil? What you do is you get a little bit of soil, add water to it, and then take a sample of that and dilute it, and then streak it out on the plate. And we did that for you. And here's the plate here. It asks the question for procedure 5A, what is the colony abundance? Give the abundance number. And it asks the question, is it even possible to accurately answer this question? And please explain your answer. So you can give a count if you wish, or you can say it's too hard, and then you got to explain your answer to give the abundance. And then procedure 5B, is there greater abundance on the colonies of the soil from the colonies of the lips? And usually students get that one right. Occasionally students have a hard time with this. Any questions about the lab? If not, that's it. I will be here until 8 o'clock, or the last student logs off, to answer questions. I doubt that anyone will be here until 8 o'clock, but I will be here until 8 o'clock to answer any questions. Are there any questions before I stop recording? All right. If you do want to ask a question, make sure you log into the lab before the last student logs off, because when the last student logs off, I will log off. let me remind people that when I share my screen, I cannot see any chat messages. So if you ask a question in the chat message, I'm going to ignore it because I cannot see the chat screen. And now that I'm not sharing my screen, I can see that somebody put something in the chat. And hopefully that wasn't a message to me. I don't even understand that message. W question. I certainly don't understand that one. All right.